Okay, so redoing this lecture for those that missed. Um, so lecture 13. So I'm not sure. Well, what we did last week, we did some lab stuff. So we'll skip over the announcements. Let's get to the content. So let's get to the content. So dairy cattle, main purpose of carbohydrates. Um, we need them to help maintain the health of the digestive tract, or one purpose. We use them for energy. We use them to build things in the body. But with our cows, we also need the carbohydrate, specifically the cell wall, to help maintain the health of the digestive tract. Without it, bad things happen. For lactating cows, we generally balance rations for a maximum of somatics, non-structural carbohydrates. They're more fermentable, so we want to put a lid on them. And then we do a minimum of NDF. It's less fermentable, and we need a certain amount to keep the cow healthy. So we want to make sure we have a minimum amount of floor for what we have. So balancing those two components, we're trying to find the optimum ration fermentability to keep the cow productive and healthy. For other animals, we're going to generally figure out the energy level that they need, and we're going to match that with the expected dry matter intake, trying to control megacalorie intake. So more often than not, we're going to have a very high NDF diet to kind of limit her intake. We talked about NDF intake as a percent of body weight, using that to challenge or control the dry matter intake. So non-structural carbohydrates for lactating dairy cow, as we said, we're going to think about a maximum as a percent of the dry matter. For most diets, most things that we're thinking about, we're doing a diet somewhere in the mid 30s as a percentage of the dry matter. So think about 35 is kind of our maximum. It can vary up to 38, 39. It could be as low as 31 as our maximum. There are no absolutes here, and it really comes back to it depends. So when we're figuring out that final number, it's more art than science. Science will get us roughly right, and then we're going to fine tune. We're going to listen to our cows. We're going to watch our cows to figure out what is right and what is wrong, and how we need to adjust. So, for thinking about the factors that are going to influence the start fermentation rate in the rumen, the fermentation rate of the NSC, non structural carbohydrates. On the left side, we have the fermentability is controlled by a number of particle size issues. So, or number of issues. So, particle size, plant type, processing, corn type, percent moisture of that starch, and the time ensiled. How long has it been in the silo? If we want to increase the rate of starch fermentation in the rumen, make it go faster, we're going to make the particle size smaller. We're going to change over to oats or wheat. We're going to process it. We're going to do something to it to change its structure. Steam flake it is an extreme example. Corn type. We gave you those two different types of corn. The white chalky material inside a corn seed that's dried down is much more fermentable than the flinty, a faster rate of fermentation. So the more we have of that, the faster things will ferment. As the moisture gets wetter, we're going to have a disruption of the starch granules. That's going to open those up for microbial attack, and they're going to ferment faster. as the corn kernel sits longer in the silo, the more acid that's produced in the fermentation soaks into there, disrupts the kernel, and makes it more fermentable. If we wanted to decrease the rate of starch fermentation, we're going to have larger particle size, so bigger. The extreme example of that is full corn kernel size. So if we want to slow down the rate of fermentation, we'll switch to corn or sorghum. Both are very resistant, higher percentages of flinty than our oats and wheat. If we want to decrease it, we're only going to crack it, not steam flake it, not grind it in some way, shape, or form. We're going to make sure that our corn has more flinty starch than floury starch if we want to slow things down. The drier it is, the slower it gets, and the shorter amount soaking in the acid generated in the fermentation, the less available it is, the slower the fermentation rate. So if you're looking at all the things that increase rate versus all the things that decrease rate, we're looking at particle size effects and structure effects. So the smaller the starch particle is, the more open, the more disrupted the starch particle is, the faster it ferments. The bigger the starch particle, the lower the surface area, the more compact it is, the slower it ferments in the rumen. So we've got to take all these factors and find that optimum particle size, type, moisture, all those things got to come together to get the proper fermentation rate of starch in the rumen. So we said somewhere around the mid 30s is our maximum, and then we adjust all those other particles to hopefully, or all other factors to adjust that fermentation rate. So we start out at, say, 35. If we have a lot of factors that will increase the starch fermentation rate, we have to decrease the amount of starch, the amount of non-structural carbohydrate in the diet to keep the cow on feed healthy and at the proper optimum fermentation rate, how much acid is produced per hour. So if we have things like smaller powder, oats, wheat, steam flaked, more floury, wetter, inside longer, we're going to decrease the amount of starch in the diet. If we have things that are going to slow the rate of fermentation, bigger particles, whole corn, corn and sorghum, cracked versus not steam flaked, more flinty, drier, shorter ensile time, all those things are going to slow the fermentation rate so we can increase the amount of starch in the diet, the amount of non-structural carbohydrate in the diet to get that optimum fermentation that we're looking for. So what we're trying to do is find the roughly right amount of things. So we do that with the science. And then we look at all these other things, the art, watching the cows, how they react to it, how are they ruminating, what's the manure look like, how much are they eating, how's the dry matter intake, all those things to figure out, okay, we're going to increase it, decrease it, starting at 35, going up, going down until we find that optimum starch level. And if we look at it, that optimum starch level, so if we look at the x-axis down low here, non-structural carbohydrate as a percentage of the dry matter, milk produced. If we have too little, we're not going to produce a lot of milk. If we have too much, we're going to get acidosis, unhealthy cows, lameness, all those things are going to reduce it. So we want to find that optimum in the middle that's going to lead to the healthiest cow, and that usually leads to the most production. So when we're doing an optimum rumen function, we're trying to balance the VFA produced, which is under the control of the bacteria, and then the VFA removed, which is under the control of the cow. So we need to help the cow manage that VFA removal by making sure the bacteria behave, to provide them with a starch mix, a optimum fermentability, so they're not overwhelming the system. That the cow can remove all the VFAs that are produced to keep the rumen healthy, to keep the pH up. So let's switch from the starches to the cell wall. So neutral detergent fiber. Lactating cows need 15 pounds of NDF per day. That works out to, with typical dry matter intakes, about 28 to 30% NDF as a percent of the dry matter. We need that NDF to keep the digestive tract working. NDF is not needed, doesn't need to be absorbed, isn't used in the body. It's only there to keep the digestive tract doing what it should. So we know that the NDF is going to feed the microbes. They're going to tear it apart, extract a little energy, and the waste product is going to be VFA. They unlock that fiber for the cow. The NDF, this cell wall, is going to provide a home for the microbes. They tend to get inside the feed and they digest it from the inside out. The long particles are going to provide that scratch factor that's going to stimulate rumination, going to keep the side walls of the rumen clean, keep the rumen toned and absorbing the way it should. Also, those fibers are going to form the rumen mat or ball that's going to form that net that's going to hold all the little particles back to get them fermented, to get them utilized to full effect. So beyond just the chemical measure of NDF, we talked about how that's done. We have to think about effective NDF or physically effective NDF. In this class, they're the same. If you go to grad school, there'll be debates on which is the better measurement. But for this class, they're the same. So we talk about the measure or the NDF that's present, its ability to stimulate chewing or maintain a balanced rumen function. So this usually means we're thinking about longer particle sizes to make things more physically effective, to make a more effective NDF. So the NDF effectiveness depends on particle size, so bigger is more effective, fermentability, fragility, and lignin content. So let's talk about those a little bit more. So increasing effectiveness, over here on the right, we have longer particle size. We have long stem hay is the gold standard when we think about that. If the NDF is less fermentable, it's more effective. So less fermentable, it doesn't get broken down as fast. It stays in the rumen longer. It can do the functions of NDF longer. So it becomes more effective as it goes through. If it's fragile, or less fragile, if it's a tough material. When we did our feed quality, how do we feel that? Is it rough? Is it break? Or is it bendy, flexible? The less fragile it is, the more effective it is. Again, it stays around the longer in the rumen. Lignin, the more lignin there is in that NDF, the tougher it is, the less fermentable it is, the less fragile it is, the more effective it is. So you can see that fermentability, fragility, and lignin are kind of intertwined. They're not mutually exclusive. We flip it over to the center column, our decreased effectiveness. We've got fermentability, or smaller particle size is less effective. So we can have NDF in our byproducts. If we think about our feed ID samples, corn gluten feed, very small particle size. That particle size is not going to stimulate rumination. It's not going to scour the rumen side. It's not going to form that mat or ball in the center of the rumen. But it's still going to be accessible. It's still going to be NDF that the animals or microbes can use for energy. but it's not going to do the other things. So we call that less effective. And then for fermentability, if it's higher, more fermentable, more fragile, less lignin, and again, those are all tied together, you're going to have the NDF disappearing from the rumen, fermenting faster, disappearing from the rumen faster. It's not going to stay around long enough to do the other things that the NDF needs to do: the scratch factor, the ball, the scouring of the sides of the rumen. So when we think about brown mid-rim corn, if you take that and you dry it like in a tray in the oven, which was what we do before we analyze it, you'll notice that when you touch it after it's dry, it tends to break apart when you touch it. It's much more fragile. It also has less lignin, and it's more fermentable, so that BMR corn is less effective than conventional corn. So again, roughly right, listen to the cows. So you're watching rumination, you're watching feed intake, all these things that suggest that the rumen is balanced in some way, shape, or form. So what we're doing, if we took all the things that decrease effectiveness, we think about mulched lawn clippings. So you've got, very soft, fragile, small, particle size things versus tough, long straw would be very effective. It's big particles, lower fermentability, less fragile, higher lignin. So, how do we satisfy the cow's need for carbohydrates? Or how do we set the percent NDF or pounds in the ration? How do we adjust? So NDF the cows need is influenced by a number of different factors. And again, we're doing the same thing we did with the non-structural carbohydrates. We're trying to find that middle in some way, starting in that middle and then adjusting by what we see on our cows, how we're doing things. So again, roughly right, listen to our cows. If we look at this diagram here, for ration NDF level, um, we can say that we start out at 30, and then if we have all of our NDF is the form of long particles, we might be able to go down a little bit on our NDF amount and still maintain health, still maintain production. If we have a lot of high fiber byproducts in the diet, we're going to increase the level of NDF, but we're not going to increase the effectiveness of that NDF. So in order to make sure we have enough long particles, the total diet is going to have to be higher in NDF to be effective enough. And then the rest is about kind of balancing the rumen, making sure that the fermentation rates are balanced and even throughout the day. If they're balanced and even and throughout the day, we feed TMR or feed grain many times a day. We have buffers and alkalizers or adequate bung space. We tend not to get slug feeding. We tend not to get high amounts of VFAs produced on an hour-to-hour basis. We can cheat down a little bit, make things a little bit more fermentable, a little less NDF. We should be able to get more production. If we've got a lot of variability or a lot of fermentability in our feeds, we need to cheat up to protect that animal. We want to make sure we add a little extra NDF to make sure we have enough effective NDF and never drop off that edge where we get into trouble. So higher fermentable in grain, higher fiber digestibility, higher variation in our feeding ingredient mixes, TMR mixes. All those things are going to lead to a bigger challenge on the animal. To protect her, we tend to cheat up a little bit on our NDF amounts. Another way of looking at the same thing, I tend to like the other one better. So lecture 14. Again, I'll put something up on Blackboard for you. Figure out what we're doing in the lab. I haven't figured that all out yet. So when we're balancing rations, we have to think about either a lack of NDF or too much non-structural carbohydrate is going to lead to problems. So as one goes up, the other goes down. So if NDF is going down, the NSC starch is going up. That's going to lead to problems. And we're dropping off the edge. We're going in the direction of a lack of NDF. So if we don't have NDF, we're going to have decreased chewing. We don't have enough to scratch factor to keep the animal chewing. That's going to lead to a decrease in salivation. The less time they spend eating, the less time they spend ruminating. We'll have lower total daily saliva flow. We assume that the buffer concentration in saliva is constant throughout the day. I don't know if that's ever been. They did a lot of work on that in the 60s. I don't know if we reconfirmed that recently or even really looked at that, but we're going to assume it's the same. So more saliva, more buffer flow. We should have or less buffer flow, less saliva, less buffer flow, decreased rumen pH. If we get increased VFA, decreased rumination or ruminal pH, we get acidosis. That is not good. Even if it's only for a couple hours, we need to avoid that acidosis if we can. So we're balancing for carbohydrates. The bulk of the volume and the weight of the diet is carbohydrates. We want to balance our NDF and non-structural carbohydrates to balance the rumen. We don't want big swings in rumen pH, and we don't want big drops in rumen pH. Spending time below 6.0 is 6.0 is considered acidotic. We want to maximize our energy intake, but maintain rumen and GI health. So you're balancing the two components, and usually, as we've said before, you're maximizing your NFC for energy intake and minimizing or having a minimum of NDF to make sure that your cows are eating the way they should. That they are healthy and protected in the rumen. So again, roughly right, and we listen to our cows. While we're here on NDF, we want to take a little detour into forage quality if we can. And one of the ways that we evaluated that was with a sensory. This is about chemical measures. So specifically, the NDF quality, how much NDF do we have and how much lignin is there in that measure. So we're looking at the fermentability, the digestibility of the NDF. The more fermentable, the more digestible the NDF, the higher the quality. So saying that same thing, forage quality, the more digestible the NDF, the higher the quality of the forage. When we're dealing with this, the higher quality is associated with lower lignin. Sometimes we can do that with the age of our feed stuff. So younger has less lignin in it, or we have a genetic mutation that lowers the lignin content. So lower lignin content, younger or less mature animal or plants. We've got brown mid-rib BMR corn versus conventional hybrids. The BMR has less lignin in it, so the NDF is more digestible. Hopefully the whole plant is more digestible because of that. You've got grass NDF digests more, but at a slower rate. So you've got the potential of grass NDF. You could get more out of it. It just takes longer. Alfalfa digests less, but what does digests at a faster rate. So that's higher in lignin. Legumes are generally higher in lignin than grass. The amount of lignin affects the digestibility of the NDF. the higher the lignin, the lower the digestibility. So these are some pictures of brown midrib corn. So you've got the brown midrib here. That's a function of you've got the plant producing the precursors or the building blocks to lignin that make things rigid and stiff on the plant. So basically, you're thinking about things in the stalk. The mutation prevents the final formation of the lignin. So the precursors or the building blocks build up and they appear brown in that midrib on the leaves. So basically, you've got a bunch of building blocks that aren't being put in the right location to do their final purpose. So brown midrib versus conventional or traditional corn silage. If you feed it to cows, you're going to get higher dry matter intake. It's digested faster, it's digested more, it clears out of the rumen, makes more room for intake. If we get more intake, more energy absorbed, the more milk will be produced. The downside is that you're going to get higher cost of seed per bag because that mutation makes things harder to produce and more expensive to produce that seed. And then you're going to get lower yield or what's called yield drag. So we associate brown midrib silages usually are about 10% less yield for the farm. So, but it depends on the soil type. Sometimes the yields are the same, sometimes they're have a greater difference than 10%. So you have to balance the benefits. High producing dairy cows will benefit from brown midrib corn, produce more milk. That's pretty standard, pretty reliable. But we don't want to be feeding brown midrib corn to our heifers. They'll just get fat heifers. There's really no reason for that. So it's helpful if we can figure out how to divide, how to separate that brown midrib from a conventional hybrid that we might end up feeding to heifers. So that's an aside on forage quality. Brown midribs are higher in NDF or forage quality. So feed efficiency, we want to talk about how much milk do we get per pound of dry matter intake. So for a lot of breeds, a lot of animal industries, this is a pretty robust measurement, and it helps us understand how much feed it takes to produce eggs and chickens and beef cows. That feed efficiency can be monitored or matched. It's usually because the animals are going in one direction as they move forward. With our feed efficiency of our cows, we have them changing physiological states. And with that change in physiological states, we're going to change our feed efficiency. And since we got a whole bunch of animals changing at the same time, the animals are not uniform, it's kind of hard to manage with feed efficiency on a dairy. You can do it broadly, but if you're trying to get down to the beyond the first decimal point, it's not going to work. It comes up every few years. People try to do it. It doesn't work. But you can, broadly say, I bought this much feed. I got this many pounds of milk out of it. There are some economic ways to look at it. But from a feeding perspective, a nutritional perspective, it's difficult with groups of animals. It's probably doable on an individual cow basis, but you don't know how much each of our animals are eating. So feed efficiency, it's the pounds of milk produced per pound of intake, dry matter intake. Sometimes we'll refine that milk pounds produced by describing fat corrected milk or solids corrected milk in some way, shape, or form. Trying to correct for the energy or nutrition in there to try to compare apples to apples. So fat corrected milk will use formulas to put everybody on a 3-5 basis, or 4% fat basis, depending on what our interests are. So feed efficiency for lactating cows depends on the digestibility of the feed and what is she doing. Is she gaining weight or losing weight? If she's gaining weight, the feed that she consumes will go toward milk production and generating fat and protein on her body. That's going to reduce feed efficiency. If she's losing weight, she's got her feed intake plus the body condition, the fat and protein off her back can also go to milk production. We're going to increase feed efficiency. So looking at that, you're going to have a situation where the feed efficiency number doesn't account for whether she's gaining or losing weight. Where is she getting the calories to make milk, and where is she putting the calories once she consumes them? So if we do by stage of lactation, we've got mid-lactation animals, fairly typical, 1.4 to 1.5 pounds of milk for every pound of dry matter intake. So these animals are neither gaining or losing weight, so this is a fairly standard feed efficiency for those animals. Early lactations or animals are losing weight. They're taking energy and protein off their back, putting it into the milk. So our efficiency goes up as measured milk produced per pound or megacalorie of dry matter intake. Late lactations are taking what she consumes. Some of it goes to milk, some of it goes to the body condition, the protein and fat on her back, and we're going to lose feed efficiency in late lactation. These numbers are rough, but if you're looking at a group of animals, you'd expect those sorts of efficiency numbers for the group if they're grouped by stage of lactation. So mid-lactations are not gaining or losing weight. Early lactation animals are losing weight. Late lactation animals are gaining weight. So again, we talked about the definition of nutrition. I define the animal, define the feeds, define the interaction. We're going to go spend some time with fats here. So fats are made of carbon and hydrogen atoms, so they're hydrocarbons mostly. There'll be a little oxygen in there in a number of different places. So fats are defined by their solubility in liquids. So they're generally water insoluble. We will also call that hydrophobic. They don't like water, and they might be soluble in ether. So not soluble in water, soluble in ether. We call it ether extract, and we'll get into how we use ether to measure the fat in our feeds. These three, water soluble, hydrophobic, ether soluble, are all synonyms in this case. What is fat used for? Well, primary thing we think of fat for is energy. As we mentioned already when we discussed calories, fat is 2.25 times the energy on a gram basis, a weight basis, than carbohydrates. Energy, it's storage form of energy for later use, and that storage form will usually predominantly be triglycerides. Triglycerides are a glycerol molecule. It's a 3-carbon chain, and then we have fatty acids hanging one off of each carbon. Those fatty acids can be saturated, no double bonds in them for those of you that have had chemistry. Almost all of those saturated fatty acids are of animal origin. Unsaturated, either one double bond for mono, or poly, 2 or more double bonds, come from plant or fish origin. What are fats used for? In addition to energy or that, we need structures and signals. So again, build or burn. So we've got cholesterol is a fat-soluble molecule. It's the basis of the or part of the cell plasma membrane. It helps keep the membranes fluid. It also is the precursor to hormones. So any of our steroid hormones, estrogen, testosterone, glucocorticoids, mineralocorticoids are all made from cholesterol. Certain fatty acids, particularly our essential fatty acids, are turned into prostaglandins. Those are form a class of hormones that go throughout the body. Probably the second biggest use of phospholipids in the body, or lipids in the body, is phospholipids. So those of you who've had biology before probably remember the uh, uh, lipid bilayer, and the building block for those is a phospholipid. So just like the triglycerides, they have a glycerol backbone. So 1, 2, 3 carbons. We have 2 fatty acids hanging off. Those are the tails you see in those cartoons of the membrane. And then on the third carbon, we have a phosphate group on there, and that is going to turn toward the water. That's the ball you see on the diagrams with the cell membranes. So all the cell membranes are composed of lipids, more or less. The phospholipids are that main building block. So that's the end for the 14th lecture. We'll go now to the lab stuff and make sure we get that covered. So lab week 7, we had feed ID, mini silos, homework due. We had a dry matter video that you need to look at from Dr. Hutchins, and then we learned how to partition the dry matter into 6 piles, and there was an assignment associated with that. It's supposed to be due at week 8, and then there are mix sheets, and there was an assignment associated with that. So reminders about the feed ID exam, that was 2 weeks. We're down to 1 week in theory. I'm not sure how we're going to do it if they close the school down, but I'll figure out something. So what is it? Tell me something about the feed. So you're going to identify the feed. So we need to get up to the classroom and study while you can. And tell me something about the feed. The questions I will ask are: what class of feed, what type of feed, high, low, high, medium, or low in NDF? Is it high, medium, or low in crude protein? Is it high, medium, or low in energy? I might ask you the reason for feeding. So if it's yeast, you're going to feed that as a additive to help rumen function. If you're feeding limestone, you're feeding that for calcium. It's the reason we feed that. So knowing something about the feed and why we feed it. Forages you're going to feed for NDF. Carbohydrates you're going to feed for energy. Protein you're going to feed for the amino acids going through. Remember our agreement going in. We want to wash in, study, and wash out. The sink is in the back corner there with a soap. So wash your hands for 20 seconds before you handle everything. Wash out so you don't take anything with you. We talked about assigned study times. The class didn't want to do that. If we're still on campus, we'll have an assigned testing time for that. So these are the bulk density calculations for our silage samples. So we had 10 samples. Each of you had one of those. So our goal is to be over 40% for a wet density. We've got some of these lower samples down toward the bottom that didn't quite make that cut. They're up there, but they're not as good as the 40s. We're looking at 14, 15, 16 pounds of dry matter. We're not making that with any regular basis. So I think you could say 3 out of the 10 samples were there. So maybe there's an opportunity moving forward if we have more packing done. We might have a little bit better smelling, a little bit stronger fermentation, get some more oxygen out of the pile. This is a picture of what we wrote on the board. This is how your feed idea will work. I'll ask you 3 questions about each feed. What is it? And then any 2 of those following questions related to that. So I need you to watch a video. We left early. I need you to watch a video, Horde's Dairyman webinar with Dr. Mike Hutchins. It's called Driving Dry Matter Intake on Dairy Farms. Control-click on the page to get to presentation mode, and then you start the video going through. I'll try to get up some questions to help you study for that as we move forward. So we've already been through the process of figuring how much dry matter feed is in a feed, how much going from as-fed to dry matter. We take the as-fed pounds, we multiply by the dry matter percent divided by 100, and we get our dry matter pounds. We've also gone the other way. If we need this much dry matter, how much does that weigh on an as-fed basis? So that's step 2 in our problem. We take the dry matter, we divide it by the new dry matter percent divided by 100, and that gives us the as-fed feeds we got to provide to that animal or group of animals. We had our calculating the new feed weights. You should have had some time to practice that with your homework assignment. Make sure that gets turned in some way, shape, or form. So what we're going to try to do here is break the dry matter down. We're splitting up the dry matter into 5 different nutrient classes. So we've already split our as-fed feeds, our wet feeds, into water and dry matter. Now we're going to take the dry matter and split that up. So to do this is very similar to our step 1, where we take our pounds, we multiply by the dry matter percent divided by 100 to get our dry matter pounds. So instead of using the dry matter percent, we're going to use the nutrient percent. So this is for protein. So how much protein is in that dry matter? We take the dry matter pounds, we multiply by the crude protein percent divided by 100, and that's going to give us the pounds of crude protein in that feed stuff or that pile of dry matter. So if we take 100 pounds of hay crop silage, haylage, dry matter, how do we figure this out? So maybe this is the best example, but we take our 100 pounds of feed, we multiply by our starch and sugar percentage of 15% divided by 100. So 100 times 0.15 is going to give us 15 pounds of starch and sugar. We multiply that 100 by 60 divided by 100, or 0.6. That's going to give us 60 pounds of NDF. So sugar and starch, NDF, are 2 carbohydrate fractions. Do the same thing with protein. 0.15 gives us 15 pounds of crude protein. 0.03 gives us 3 pounds of fat. 0.07 gives us 7 pounds of ash. So sugar and starch, NDF, are going to represent the carbohydrate. We have crude protein, protein fraction. We have our fat. We have our ash as our crude minerals. And we already know the water in the feed. So that takes care of 5 different classes. The one we're not going to do here is vitamins. Vitamins so small, it's almost noise in these calculations. So it's really not a significant weight as we move through. So let's take a ton of feed and work this out on the whiteboard to figure out, in a pile of feed, how much do we have of water, of starch and sugar, NDF, protein, fat, and ash. So this is what we did on the whiteboard. It's a 2-step process. So the first thing we do is take our as-fed, step 1 in the green here, we divide it into water, so pounds of water and pounds of dry matter. Then we take, in step 2, the dry matter, and we divide it into our 5 large nutrient classes: crude protein, our 2 carbohydrate classes, fat, and ash for minerals. So if we take 2,000 pounds of hay and do fairly typical measurements, we have 2,000 pounds of hay. We say it's 12% water and 88% dry matter. So 2,000 pounds times 0.12 is going to give us 240 pounds of water in that ton. And then if we take 2,000 pounds by the dry matter percent, 0.88, that's going to give us 760 pounds of dry matter in that 1 ton of hay. We move on to step 2, where we take the dry matter, so that's 1,760 pounds, and we multiply it by the percentages associated with each fraction. In this class, I will try to make things add up to 100. That will not happen in real life all the time. It'll be close, a little over, a little under, because of the analytical errors associated with getting these numbers, but I will try to make them add up to 100 in this class. So 0.1 times 1,760 means that in that pile there's 176 pounds of protein. 15% sugar and starch, multiply that through, we're going to come up with 264 pounds of sugar and starch. NDF, the biggest fraction, we've got 65%, we've got 1,104 pounds of NDF in that 1 ton pile of hay. 4% fat's going to give us 70 pounds of fat, 6% ash, 106 pounds of ash or minerals in that pile. So if this column over here, our protein, starch, and sugar, NDF, fat, and ash adds up to 100, the weights over here have to add up to that 1,760 pounds. So that's a way to check your math to make sure you did everything. So step 1, you should add up to the total you started with. Step 2 should add up to the amount of dry matter that you had. So that's a 1 ton pile of hay. We go over here and 2,000 pounds of corn silage. It's 65% water or moisture, 35% dry matter, 10% crude protein, 37% starch and sugar, 46% NDF, 3% fat, 4% ash. You should be able to go through, follow steps 1 and 2, and generate those numbers for yourself. So pause this video and give that a try. The other thing we did in lab was making mix sheets. So when we balance rations, we do that on a dry matter basis and on a 1 cow basis. Up at the dairy, when we feed cows, we're doing a pen of cows, and we're doing on an as-fed basis. So what we have to do is take that dry matter mix for 1 cow and convert it to as-fed pounds in a mix. So if we look at the ingredients that we might be using up at the dairy, we have the dry matter pounds that the ration calls for each of these. So 20 pounds of corn silage dry matter, 9.65 pounds of hay crop silage, 2.55 pounds of canola meal, 7.65 pounds of lactating cow grain, 8 pounds of cornmeal, 0.2 pounds of protected fat. We'll assign the dry matter percentages for each of these, and we need to go from dry matter pounds in this first column on the left to the as-fed pounds per cow per day in the middle column. So to do that, we take the dry matter pounds, like we did in step 2 of the finding the new feed weight. We divide by the dry matter percentage, and we get a larger number because we're adding the water back in of the as-fed pounds required per animal. So we take that, and I better make sure I screwed up in lab, make sure I get these right. Let me check on a calculator here for a second. Would help if I turned it on. So 57.1 times 118. Okay, so that looks approximately right. If I carried out more decimal points, I'd probably get that. So you're looking at a number somewhere around, if we multiply by 118 animals in the pen, we get how many pounds we need to include in the mix. If you've ever mixed feed, you know that the nearest pound is usually a joke. We go to the nearest 10 pounds. So somewhere around 6,740 pounds of corn silage, 3,000 pounds of hay crop silage or haylage, 340 pounds of canola, 960 pounds of grain, half a ton of cornmeal, and then about 24 pounds of half a bag of protected fat gets the job done. So mix sheet, depending on if you do this continually or you round things off, you might get a slightly different number than we have there, but as long as you're within a few, that's good enough. So that's how you do a mix sheet. So this over here in the right-hand column is the mix sheet. That's what you're going to use to generate your mix and deliver the cows. So there's a homework assignment with partitioning up feeds. So that's one assignment. And then there's 2 mix sheets that you have to generate for the current rations up at the dairy. So those are posted on Blackboard. Try to get those done. If you have problems, contact me some way, shape, or form. We'll make sure that everything is where it needs to be.